cells.
What is up, everybody? Welcome to another episode of The Rabbit Hole. I'm
Rabbit hole.
Ben,
I'm Ashley. Hello.
and today we're going to talk about biology, about the cell specifically. We talked about it last week, and we're going to do another deep dive into the microscopic levels of the universe. So I am ignorant of a lot of the stuff about cells. I know that the mitochondria is a powerhouse of the cell because, I mean, it's classic. It's
Classic. Like if you take anything away from biology, most people will know that the mitochondria is the powerhouse indeed of the cell.
Because, and also it was a meme. I don't know if you've seen the meme, but
Always, yeah.
memes, yeah.
And it's totally worthy. Like the mitochondria deserve so much love and respect. So I'm glad that the internet is at least giving them.
Okay, so yeah, I would love to.
They're actually the ones that powered the ability to do that. They kind of deserve it.
Okay. So yeah, I would like to know, and just kind of an overview of the cell, what makes up, and this I know at least that there's a difference between animal cells and plant cells. So we can just talk about like the animal, so if that's a good classification for it, and then
Yeah.
some of the basics of that, and then we can dive into the mitochondria potentially because it needs the attention apparently, and then cell metabolism.
Yeah, totally. I mean, the biggest things between animal and plant cells overall is that plant cells have cell walls, and that's where you get things like if you look at plants, sometimes they're like droopy and they're all fallen over, and then you water them and then they come and they stand back up. That's because of the cell wall, which is really cool. And so when you give a plant water, you get this thing called turgor pressure, and then it makes the cells become more rigid again so that the cell can stand up or the plant can stand up. And then also, obviously, plant cells have chloroplasts, which allows the plant cell to perform photosynthesis, which we are unable to perform because we do not have chloroplasts.
Now, are there any other types of cells?
Between, animal and plant, animal cells and plant cells are the two main ones we talk about. Yeah.
Okay,
But
now
I mean, but those are eukaryotic cells. Then yeah, of course there are bacterial cells and there are archaea.
that's what I was curious about. Like, how do you classify bacteria? And then I know viruses are kind of special.
Well, so viruses aren't cells. They're like we haven't
I've
really
seen the picture of them, and they look like something off of like the Matrix.
Viruses are wacky because they meet some of the criteria for life and they don't meet some of the criteria for life, right? So a key thing of viruses is they need a host. They need somewhere to replicate. Now they do have their DNA and they inject that DNA and kind of hijack the host so that the host does all the work for them, which is amazing. But yeah, so there are three kind of classifications of life. There's the prokaryotes. Well, these are the two main ones. There's prokaryotes and then eukaryotes, which I believe we talked about last week. The eukaryotes are characterized by the membrane-bound organelles. And then there are, within prokaryotes, there are bacteria and archaea. And archaea are these like crazy resistant organisms that are able to withstand insane temperatures and really like abrasive conditions. And these are the types of organisms that you'll even find living in a volcano.
It's kind of like the roaches of the roaches and the mice of the cellular world.
They are durable little suckers. And so there's them, and then there's eukaryotes. And then within eukaryotes, that's where you get into like fungi, animals, plants, kind of all of those different kingdoms, but the main domains of life are the bacteria, archaea, and then the eukaryotes, which we are eukaryotes.
So what's prokaryotes?
Bacteria and archaea, and they have no membrane-bound organelles.
Gotcha.
And their DNA isn't, I mean, they still have a nucleus and everything, but they were probably the beginnings of life, a really, really long time ago.
So we're talking about I want to dive into the eukaryote side and into that out of the multiple branches, the specific animal side of that. So that is what I would like to kind of dig down into on that one.
Okay, then let's do it. That's the one that I'm the most versed in. That's the one I spent most of my time studying. I still know a bit about plant cells because we had to learn about those in cell biology. and learning about how those cells complete respiration and how they use the chloroplasts to harness the energy from the sun and the photo conversions that go on so that they can and there's it's amazing. We could actually do an episode on that too. It'd be really cool to kind of walk through photosynthesis and explaining how it
Oh, that'd be great. That'd be great. Because I mean, I haven't done it in since like middle school, and that version I'm sure was simplified greatly. And so I think
yeah,
it'd be really nice to expand it and really get a more a better grasp and understanding of photosynthesis.
that would be so fun because we can break it down and really go through how it works. And there's all this stuff with like electron cascades, and it's just an unbelievable process. So yeah, let's do that. That could be like just a short and sweet 20 or 30-minute one that I just talk through the process and
Hmm.
for anyone who's interested in photosynthesis.
Hmm. Sweet.
Which is amazing that plants have this ability to literally harness the sun's light and turn it into energy. That's pretty cool, I think.
And I think the one thing that's amazing is so many innovations that people create are merely imitations of what they've already seen in nature.
Totally.
And
Yeah.
so even flying, I read or I listened to this great audiobook. It's called The Wright Brothers. North Carolina claims them. I'll take it. And
It's even on our license plates.
sorry, Ohio. And in that book, whenever they were running tests and stuff, they got inspiration from birds. I feel like men have wanted to fly forever. Icarus, like Greek mythology, and
Great theory.
Hermes with wings on his feet. All this, everyone wants to fly, and so they started modeling their airplane after birds and really trying to understand the anatomy of birds and how they could fly. And from that, mechanical machines have been created. In the same way, I think of like solar panels, and people are like, okay, somehow plants are getting energy from the sun, and there's so much energy here. How can we do this? And like we know it's possible. Nature's done it. So I think that's such a cool inspiration to draw from nature by learning this stuff. So that's another reason why I think it's awesome that people know so much about biology.
And even talking about flying, I don't know about you, but every time I see a bird in flight, it's just like the sense of awe. Like you just watch it and you're like, wow, that's so amazing. Or especially if it's a bigger bird. Like I've just been seeing a lot of really big birds lately.
Yeah.
And there's something about it that it just like it demands attention. Like you just have to look at it for a moment.
Yeah, I was really close to I think it was a Cooper's hawk today. It like flew right in front of me, and it's just yeah, it's very impressive. I mean, most of the time, um, I don't have too much awe, but then when I really think about it, which is why this is another reason why stuff like this is so good, is because it does make you appreciate and take your and pay more attention to things that are going on around you that I don't see all the time.
I remember in college when I was in my like biology class and I stepped outside and we had just done the photosynthesis lecture for the day. And it was on the Brickyard, right? Because I was in Boston and the Brickyard, there's so many trees. And I just walked out and I remember just feeling in this like I had entered this new world, like the doors had opened and Terabithia was there. And I was just like, this is so crazy because this is what these plants are doing. And then they're providing this oxygen, which just happens to be the byproduct of their metabolism that then I need. And then my byproduct is carbon dioxide, and they take that in to fuel them. And there's like this kind of neither can exist without the other. And it's just like so beautiful. And then you take that and all this biological knowledge and you go into the forest, and it's just like totally awestruck because everything around you is helping you live and you're helping it live. And that's so beautiful.
Yeah, it's I think that's one thing that's amazing about being educated. Now, there's some serious downsides to education if it's not good education. You can be, indoctrinated, and it's really hard to break those uh, molds. But in its best form, the humanities, that's what they're trying to do. They're trying to tell you to focus on certain things that really brings a lot of joy out of life, And I remember seeing an Instagram picture. It said how most people see colors, and it had like, blue, orange, red, green, and then it had like how artists see colors, and it was like, grrrr. And so yeah, I want to see what the world, like how painters see the world, like what they notice, uh,
Right.
and how, like, so I've been surveying, and every single time things catch my eye because that's what I'm trained to, like, every time I see pink flagging or something, it just, or something on the ground, my eye just goes to it. It just happens. And so how much does that affect you of what you've been trained to see and manifest itself? And you can, yeah, you can be trained to see the beauty of things, you can be trained to see the negativity of things, and not to be ignorant of either one, but yeah, that's really powerful. So
Yeah, it really, like you said, contingent on everything being good and it being quality education that's as reflective of the truth as possible. It just makes everything else so enriched, I
mhm.
think.
Yeah. Oh, so back to the cell.
All right.
Sorry, you took that tangent on there, but
No, no. I at all biology just always opens the doors. But so we're wanting to talk about specifically animal cells. And is there anything specific you would like to know, or do you just want me to kind of start talking, or
yeah. So the way I see cells, I'm gonna give you my best shot, and then you can fill in the gaps. Maybe
I'm
that's.
ready. You be the teacher for the next couple of minutes and you just tell us.
All right. So let me tell you about the cell. One thing you got to know about the cell is the fundamental building block of the human body. Um, it's really important. Without the cell, there would be no life. Now, enough about that. If you could take a look inside the cell, you have the nucleus, you have nuclei, you have the mitochondria, which is the powerhouse of the cell. You have riboflavins in there. Um, and then you have the Magellans. You also have the neuroplasts. You have, which I'll get I'll get into this later. You have the cellular membranes.
Very good. There you
And,
go.
and one thing that's interesting about the cell is it only lets some things in and some things out, which is very
Ah,
important. It's really
collective little
fascinating once you
fuckers.
get into it. Let's see. Um, and it multiplies in a mysterious way. Um, and some cells are different than others. You have the red blood cell, white blood cell, um, which you have lots of things. And so that's basically what a cell is. All right, now, would you like to tell us what an actual cell is comprised of in a much more articulate way?
I don't know. I think you just did a great job. We can end here.
Yeah, those white blood cells, man.
Ah, the immune system is such a beautiful thing and it's amazing. Like you actually have cells that do antigen presentation and they go and they like after they've been infected with something, after you have an infection, they chomp it up and they find the defining part of the whatever disease it is you have or virus or bacterial infection or whatever and they bring it and they actually like present it like hey we need to remember this for later and then it gets incorporated and that's how vaccines work right like you're introducing this to your immune system so your immune system says oh okay I recognize this therefore when I see it later I don't have to go through the whole process of reinventing the wheel I already have the powerhouse and the kind of like reserves to say oh I've had this before I know this now get to work go get rid of it which is just unbelievable that like our cells are kind of like they have this memory they have memory and they know what they've encountered and what they haven't and so you have these that you have specific immune cells in your immune system that are memory cells which is unbelievable. I remember like every single day going into the lectures for the immune system my jaw was just on the floor I was just sitting there like dumbstruck I couldn't even take notes I was just so like what it was amazing. um but yeah so you're totally right every cell has a nucleus. The mammalian cells they have the nucleus and that's where your DNA is stored and it's super protected. Your DNA is precious. And I think I talked about this last time, but every cell is biologically equivalent or genomically equivalent. So all of your cells have the same DNA in them, but it's kind of the buddies that they're around or their differentiation path that they've gone down that tells them this is what type of cell I am. So that's why you don't have a liver cell in your lung, unless you have like a metastasis. But so there's the nucleus, which holds the DNA, ribosomes, which we talked about, I think, last time, which
which
help
I,
make protein.
which I said was riboflavin, which is
Yeah,
as well
it's
as
a it's a compound, but ribosomes, it's you got the ribo part, so that's you got that going for you.
sweet.
And they make the proteins, so they translate the mRNA that you've transcribed from your DNA, turned it into this RNA language, and then that gets translated into the protein language. And then you've got the Golgi apparatus, you've got the endoplasmic reticulum, you've got the mitochondria, of course, like we talked about, and there are several other organelles that are floating around, but those are kind of like the main players that most people have heard of. Proteasomes, which chew up proteins and help recycle amino acids throughout the cell. So there are all these kinds of all different organelles. But the mitochondria, and the cell membrane, which I think is like really overlooked because we spend so much time talking about the mitochondria, but the cell membrane is amazing. And it, like you said, it is selective. So it's called the fluid mosaic model. And it's because it is fluid and it flows and proteins move around throughout the cell membrane. And so in the cell membrane, that's where you have different types of like channels or carrier proteins, there are different types of proteins at the cell membrane. You can either be peripheral, which means it's just attached to the kind of the periphery of the membrane, or you can actually be transmembrane, and those actually span the entire width of the membrane. And those are like the channel and carrier proteins. So channel proteins are comprised of units that allow a specific ion to enter. So they have specific affinity for maybe a sodium ion or something, where the inside it has the polarity and the affinity for a specific ion, and they can just readily flow through these channel proteins. Carrier proteins, they actually are in an inactive conformation, so we usually talk about it like this, but then when the correct substrate binds, it changes conformation, and that lets it go into the cell.
Wow.
So it's inactive, and then anything else that'll bounce off of it, right? Like everything's kind of bouncing around all the time in the cell. Will just keep until the real thing that's correct that you want to bind binds, and it has a specific affinity. So this is more biochemistry where you get into like enzymatic reactions, and it's called a specific type of plot, and it actually looks at affinity between things and then kind of how much more likely their binding gets when they're brought into the same vicinity because of the conformation changes that start happening. But yeah, so the thing will bind and then it'll change conformation and it'll get dumped into the cell. And then if it's no long after that happens, it'll return to its resting state. And so a big one we talk about with this is the sodium potassium pump. Did you learn about the sodium potassium pump? So the sodium potassium pump is a way.
It still has to do with the cell membrane of letting things in and out. So we're looking at the cell membrane, and how it determines what to let into the cell, which they need to be very careful about, because if they let wrong things into the cell, it could corrupt the cell or kill it or change something.
Right. It's a very strict game about what goes in and what goes out. And so cells, a big thing that we always are talking about is affinity to or polarity. So the inside of a cell is negative with respect to the outside. So with respect to the outside, you have a negative, the inside of the cell is negative. And then you talk about things in their gradients. So if you're looking at specific ions, maybe there are more ions inside the cell than outside the cell. So if those ions try to go inside the cell, right, it's kind of like solutes. A solution can become saturated and then it can't take any more solute. So you can kind of pull on some chemistry topics to think about this. But if the ion concentration inside a cell is higher than outside the cell, you're going to have to use it's going to be active transport to bring that into the cell, because it's going against its gradient. It's going from low to high, right? And it's naturally go high to low, right? So If you're bringing something in against its gradient, that requires active transport. So the things I was just talking to you about, like channel and carrier proteins, for example,
That's
most of these are
passive.
passive transport.
That makes sense because it's waiting. This thing, the stuff is moving from high to low energy. It doesn't require any outside.
Right. You don't have to pay with this currency. And the currency we really talk about the most with cells is ATP. That's adenosine triphosphate. And so it's adenosine attached to 3 phosphate molecules. And phosphates also help power a lot of things of the cell.
That's where the energy is stored in this molecule.
ATP is just like an energy hotspot. Yeah, it's just energy, energy, energy. And a lot of reactions, and why it's called the currency of the cell is because many reactions need ATP to drive the reaction.
That's pretty cool. I like because in mechanical engineering, we pretty much never talk about chemical energy. Ours is mainly energy from because I mean, a lot of these equations in physics are like you make assumptions, and so for us, we would pretty much always assume that the chemical energy would be like 0, or they would give it to us, or we'd figure it out in some type of chart. And so that was like, well, like okay, it's 10 watt, it's something large, uh, and then we would work on like heat loss from like friction. That would be the energy.
Right.
And so that's so cool hearing like the chemistry biology side of the way you use calculations for energy for ATP and like there's so much, and then yeah, there's so much energy stored within um, compounds, molecular compounds. It's really cool.
Oh yeah, so much energy. And we talk a lot about it in terms of like Gibbs free energy. And like that compares, right, like entropy, and looking at we always want entropy to be positive. We want things to be going towards that state. So so ATP is the currency, yes. So the sodium potassium pump, it's in this kind of inactive
Is it
conformation.
in the cell?
The sodium potassium pump is on membranes.
Okay.
It exists on the membrane. And then it's in this conformation. And then sodium will bind. It will bind 3 sodium ions.
That makes sense. Sodium is readily available, or to, yeah. Okay.
And then once those sodium bind, it'll change conformations. So we go now to this conformation, and then the sodium gets dumped into the cell. Okay, so we're moving from outside the cell to inside the cell. So we're like this, sodium bind, dump them in. Now the potassium from inside the cell is going to bind. So we get 2 potassium, and then we switch the conformation and dump the potassium.
So well, how come? So 2 potassium go in, but what if they just touch right back and they go back out?
If they touch,
So you said it's like this: 2 potassium hit the membrane in this sodium potassium pump.
right?
It goes boop, and so now it's floating within the fluid or whatever is inside the cell. But what if they just land right back, and then you activate it again with
Because good question. Very good. Yeah. Once the potassium are dumped, it changes back. It changes conformation back. So they can't bind.
So it goes like this.
Yeah. It's like constantly like this.
Okay.
It's this game of just constantly opening and closing to transport. And so that is all about bringing things into the cell and an active transport, but that requires energy because you've got things that are going against their gradient, and whenever you have that, that is active transport. So how we get active transport is for active transport to work, you have to pay, and you pay with ATP.
The currency of the cells.
Currency of the cell. If you take anything away from this, ATP is the currency of the cell.
I have a question though.
Okay.
Why do you need to bring anything into the cell in the first place? What is the goal of bringing something into the cell? I'm guessing food for some energy. It needs energy to survive.
Yeah, and cell communication, cell signaling. So you have to maybe get something into the cell. You need to send a signal. You need sodium to act as sodium can sometimes act as like the start of a signal cascade or a transduction, and tell the cell, hey, look, it's time to go.
I would also like to take it a step further back so I can understand this a little bit better. So I understand that atoms are extremely small, and then you have molecules, which are collections of atoms.
Right.
But how big are cells? And are there different types of cells? I know, like we talked about, like you said, liver cell, heart cell, um, all of that jazz. Like, how big are these cells we're talking about compared to like atoms or molecules or something like that?
Yeah, so cells are way bigger, like way bigger, magnitudes of difference bigger than atoms and molecules because cells are a collection of
Oh,
all these molecules of organelles, right? Like so I mean, cells are microscopic to us, but cells are ginormous compared to molecules and atoms. Because a given cell, I don't know how many molecules it has packed in it, but a lot. It's a big number. So even though cells and what's crazy is even if you take a cell and just look at it through a normal microscope, like you really only see the nucleus. You don't see all the other organelles. It's not like you look through a microscope, like a normal microscope you would find in a lab, like not a
yeah.
specialized.
Yeah. So when you're looking at a cell in the lab through a microscope, um, so um, what does like a red blood cell look like to a liver cell, and then what do you see when you're looking through the microscope? You only see the nucleus. Do you see the membrane? Do you see the other organelles? And also, what's the fluid or whatever that's just spinning the organelles with inside the cell?
Yeah, so you won't see any of that. So like when I was at my job and I was doing my work, I was using HEK cells, which are human embryonic kidney cells. And yeah, it's just a lot of them. So you see a bunch of cells, and if you really zoom in, you could maybe get a glimpse of the nucleus, but we're just talking a dot, a blob. It's not like the well-defined nucleus that you see in the science textbooks that looks all beautiful. It's not like that. And even what's crazy is we have to think about like the diagrams are what you might envision in your mind as a cell. Like the cells are composed of this amazing thing that's called the cytoskeleton, and it's how everything kind of ties together and what actually organelles can be suspended and attached to. And the cytoskeleton is a bunch of different fibers that run all along the cell, and
Ah,
that things can,
inside the cell.
yeah, if this is inside the cytoskeleton, just like we have skeletons, cells have skeletons.
Ah, that's cool. Because when I look at the textbook, it's like a purple cell
No,
that has like these organelles floating around that are cut in half, and
right.
it's like planets floating in space. That's the image that in the textbooks that I imagine.
Right, and that's what I'm calling on is like think about that image and then just think about how wrong that image is because the cell has so many filaments that are running along it as the cytoskeleton, and then that is the these are like railroads and highways for the cell. So you have these amazing things, and maybe one time we can do a share screen and I'll like show a video of it. But like motor proteins are unbelievable. So motor proteins literally have legs and they walk along the cytoskeleton
That's cool.
like one foot in front of the other, and it's all about polarization and like forming these bonds so that they can walk, and every step requires ATP, so this is a very active transport, so this is a high investment, but it's amazing. They have legs and they kind of have arms, and they'll carry like a vesicle or something, and the vesicle will have a tag on it that tells the motor protein this is where we're going, and then the motor protein knows how to get that cell that to where it needs to go. So like a common one is ubiquitin. Ubiquitin is like a common universal signal. It's actually one of the most highly conserved proteins in the animal domain, which basically means that between all species, it's one of the least changed proteins. So that signifies that that protein is crucial, right? Because the less changes something goes through, the more you know, like, oh, this has to be a mega player, and ubiquitin is.
Oh, just a little side note. I was listening to a Joe Rogan podcast. I think the guy's name is Forrest Galanti. He's a biologist, and he goes around, not molecular, but he goes around and yeah, I guess a biologist. Yeah, he goes around and finds animals that are presumed to be extinct,
Yeah.
and they're talked about alligators and crocodiles and how they're like, yeah, they're pretty much like the hammers of the animal world, like, super simple, doesn't need to be changed, good to go. It's a tool that just works, and it's so simple. Like, an alligator does not change, it literally just sits by water, eats. It's like it serves its purpose. It's like a hammer. Like, it's so simple, it doesn't need to change, and it's survived so long. And so when you said that ubiquitin, I was like, yeah, it kind of makes sense, even for animals, you know.
Yeah, and ubiquitin is such a unique protein because it gets tagged to anything really and tells it, oh, this needs to be degraded. Get rid of this. This is trash. It's like a trash signal to go to wherever it needs to go. Um, and then the motor protein will walk along, and it's unbelievable what these motor proteins do. Like they're so cool. I've watched so many videos on them, and just it's like I'm telling you, they have feet and they walk like step by step, and every step requires active energy, and it's just so amazing to watch. But so yeah, so that's just another example of we've got to pay. It's an upfront cost, but this vesicle has got to get here, so we have to put in that investment.
Okay, so
So
I'm starting to get the picture a little bit more. So there's a cell, it's full of organelles, all connected through the cryoskeleton,
cytoskeleton, yeah.
cytoskeleton.
The
And
prefix cyto means cell. So anytime you hear cyto, you know you're talking about a cell.
okay, cytoskeleton. And the cell, it's one of its jobs is to, you know, obviously continue propagating itself and like cell, would you say cellular metabolism, or is that basically when it breaks apart?
Metabolism, respiration, yep.
And there's other functions.
Apart? No, that would be like the cell cycle, like
Stuff
mitosis.
like mitosis.
Yeah.
So yeah, and every cell serves a slightly different purpose, but they're all relatively similar when it comes to their nucleus. They all have the exact same information in the nucleus. Is that right?
Yes, they are all genomically equivalent. They all have the same DNA. It's just depending on what DNA is getting expressed. Yeah.
And so in order for these cells to serve their purpose, which it depends on the cell, and to uh, um, you do mitosis, then they need energy.
Perfect.
And
Beautiful.
so in order to get energy, they they get it from the outside somehow. From how do they get the energy? You know,
So
like
energy comes from food, right? Food is literally energy because your food, so
McDonald's.
so digestion starts in the mouth.
Hmm.
You like we think of digestion happening in the stomach. No. Digestion starts in the mouth because you have different enzymes in your mouth that are going to already go ahead and start digesting food. And then it goes down, right? So it goes through this long, extensive process, and that could be a digestive system podcast, but ultimately it gets broken down into its smallest components, and that's the macromolecules. So, right, you've got carbohydrates, you've got like if you're broken down into carbohydrates are that the core of carbohydrates, the core unit of a carbohydrate is glucose. And so we get broken up into glucose, and then glucose is kind of the simplest pathway. Now, all kinds of foods can enter this cell respiration metabolism pathway at different points, but like there are different entry points, but we're going to just start with glucose. So glucose, have you heard of glycolysis?
I have not heard of glycolysis.
Okay.
Sounds like a disease.
So glycolysis, so this is all the beginning phase of forming ATP. So glycolysis, we're going to take a glucose molecule and we're going to blow it apart, pretty much. Blow it up. And this actually happens in the cytosol. So glucose just comes into the cell. Like glucose can just diffuse across the membrane because the cell knows I need glucose desperately.
Okay.
So we don't want to expend energy getting glucose in. We just want it to be a free flow and highway. So glucose just comes in to the cell, and the glucose comes in. And then we go through a series of processes called glycolysis to blow that glucose apart. So there's a lot of conversions. We've got to get it into different forms. We've got to blow it apart.
There's energy stored within glucose within the chemical bonds, and you're blowing that up to get and through that process, energy is being released, and the cell is somehow transferring the energy stored in glucose to with ATP. I know that's the currency somehow, but and it's using that energy to do its thing.
Well, glycolysis is really the precursor. So from the glycolytic process, you only get you ultimately in total you produce 4 ATP, however, per glucose molecule. However, you have to invest 2 ATP to blow it apart because glucose is such a stable molecule. So you have to have this upfront cost. So
Ah,
the net of glycolysis is really only 2 ATP, which definitely isn't enough. We need loads. So glycolysis is just the first step. So the whole point of glycolysis is take glucose and turn it into pyruvate. Pyruvate is a molecule that will then go on through this complex, and it will be converted into this thing called acetyl-CoA, acetyl coenzyme A. And acetyl-CoA is crucial to enter the Krebs cycle or the citric acid cycle. There are lots of different names for this, TCA. But Krebs is what I'm going to call it because Krebs is the person who delivered it or discovered it. And this is a relatively new discovery. I mean, I don't think we even knew about this 100 years ago. Oh, this is pretty new. So Krebs, acetyl-CoA comes in and it joins with the first molecule, and we form it joins with this thing called oxaloacetate, and we form citrate, which is why it's called the citric acid cycle, because you form this molecule called citrate. And that citrate is going to go through a series. The Krebs cycle is a circular thing. Okay, so you make oxaloacetate, combine that with acetyl-CoA, get citrate, run Krebs. Oxaloacetate, acetyl-CoA, run Krebs, etc., etc., etc. Krebs is this continuous cycle, and this is happening in the mitochondria. So now we're in the powerhouse of the cell, and now we're going to learn why. So we're running Krebs, and the whole point of Krebs is not to produce ATP. The whole point of Krebs is you're loading up these things that are electron transporters. You can think of them like taxis. You load them up with electrons. There are these two, there's NAD and it's technically NAD plus, and then there's FADH. And NAD plus and FADH, if you know anything about chemistry, the big thing is oil rig, oxidation is losing, reduction is gaining. And so these are compounds that will be reduced, meaning they gain an electron. So they are electron acceptors. They are able to become more negative and to take on an electron. So think of NADH or NAD plus and FADH as these electron acceptors.
and this is happening in the mitochondria?
This is in the mitochondria.
And this is a process of the Krebs cycle? This is a part of the Krebs cycle?
Yes, and this is happening in animal and plant cells. This is happening in you. This is happening in me. This is happening in Sirius. This is happening in the plants in my corner all the time.
So what's getting put into the cycle again? Still glucose? Or
well,
the precursor, glucose was the precursor to get something, and now we're getting that. Now how does the glucose, okay, it's coming into the cell membrane, right? And of course the cell lets in glucose because it already knows you've got the toll pass, come on in.
Yep.
So it comes in, now it comes in, it's in the cell. When it looks around, all it sees is a bunch of cytoskeleton. It doesn't really know where to go. How in the world does that glucose know how to get to the mitochondria? Or does it hop on the backs of these protein walkers that take it to the mitochondria?
Okay, so the enzymes that you need for glycolysis are just present throughout the cytosol, which is like the jelly-like substance of the cell.
So the cytoskeleton and cyto.
The cytosol is what everything is suspended in. It's just the fluid of the cell. Like we have blood. It's like, obviously it's not blood, but it's kind of like, just like we have a skeleton in blood, the cell has a cytoskeleton and the cytosol, which is just the liquid that is filling the cell. Yes, Ben.
So plants have plant walls. I'm imagining that it's like an exoskeleton. Does these plant cells also have a cytoskeleton?
Yes. Mm-hmm.
So inside the cell, you got the cell, the membrane, and inside the cell membrane is the organelles, and you have the cytoskeleton, and then the cytoslime.
Oh, cytosol. Yep.
Cytoslime.
Sure, that works too.
Cytosol. Okay, so and inside the cytosol, you've already got enzymes that are active and ready to break down this glucose. So you're almost getting like a soupy mix of goodies.
Of goodness, yeah.
Of goodness, and it's all in the cell. So now it's kind of just floating around in the mitochondria inside the cell. It's basically like a little pond that it's already got the stuff it needs, and it's kind of taking in these enzymes these enzymes from the glucose after the enzymes have already broken it down, which you said that process was something
Glycolysis.
glycolysis.
So think about it, right? Lyse means to lyse, to break apart. Glycolysis, lysis, breaking apart glucose.
Okay, that helps.
See, the clever thing about biology, everyone's like, oh, big words, but the words tell you so much about what's going on, which is amazing. Like even the citric acid cycle, citrate, boom, citrate, boom, like it's a cycle. So we've gotten the glucose, and we've blown it apart, and it's now in pyruvate, and pyruvate becomes acetyl-CoA, acetyl coenzyme A, and then acetyl coenzyme A joins with oxaloacetate, and we form citrate, and we run Krebs. Whole point of Krebs is to load up these electron taxis. NAD plus becomes NADH because it's added an electron, and FADH becomes FADH2 because it has also gained an electron. So all throughout Krebs, all you're doing through the series of converting the molecule is all of that is to release electrons from that molecule and give them to these electron transporters. So these electron taxis, when I TA'd this class, I always just had a taxi come up. Like whenever I talked about them, I had a taxi fly in on my slides because I was like, anything you need to think about is just a taxi. You need to think of this as a taxi that's then going to take these things that we've loaded up from Krebs cycle that have the electrons. So I'm just reemphasizing that the point of Krebs is not to produce ATP. It's a step in the way of producing ATP, but Krebs itself is about loading up these electron carriers that are going to go to the electron transport chain, the ETC. This electron transport chain is embedded in the membrane of the mitochondria. So just like our cells have a membrane, the mitochondria has a membrane. And the electron transport chain is all based on electronegativity. And electronegativity is how grabby is the molecule for electrons. How badly does it want electrons? So the grabbier it is, the higher the electronegativity. And the electron transport chain is beautifully crafted because every molecule in the chain is successively more electronegative. So that pulls the electrons down the chain. Because this one so the first one in the chain, the first complex in the chain, accepts the electrons from one of the carriers. And then the one next to it, the complex next to it says, oh, wait, no, I want them. And then the one next to that, oh, no, I want them. And it keeps getting pulled down until we get to oxygen, which is the most electronegative and the final electron acceptor of the electron transport chain. So now what's happening though, all along as these electrons are moving down, it would be super helpful to have a visual, but kind of hopefully you can sort of get a grasp of what I'm saying. As the electrons are moving down the electron transport chain, we are pumping hydrogen ions outside of the membrane of the mitochondria. And why that is because at the very end of the electron transport chain, there's this thing called ATP synthase, and it's literally a rotor. It rotates. It physically rotates. And it is powered by these hydrogen ions that have been pumped out. And as those flow down their gradient through the ATP synthase, we do a revolution and we form a molecule of ATP.
So is this happening on the membrane of the mitochondria, you said?
Yes.
Okay. So
yes.
inside the mitochondria, yeah, it's you said phrase it as they're creating NHP positive or something, and they're taxing they're taxing on top of the membrane of mitochondria, and they're going down the ETC. They're going down the electron transitivity chain,
etc. Yeah.
And now after all the hubbalaboo of finally the oxygen atom gets the oxygen atom gets what's his, and after that, and they're releasing hydrogen ions somehow. I'm not quite sure about that yet. ATP is formed at the end of this on the membrane of the mitochondria.
Yep. And so the hydrogen ions are pushed against their gradient after the NAD and after the NADH and the FADH2 have dropped off their electrons. And all like we can get into more specifics about that in a next podcast because that's a little bit I feel
Yeah.
like a diagram it would be more helpful. But they're pumped up here and then they're gonna come down over here at this ATP synthase and they're just gonna flow down their gradient, which is the inside of the ATP synthase. As they flow, the ATP synthase will rotate and we're just gonna crank out ATP molecules. And you get like the number is kind of debated, but it's somewhere from 32 to 40 ATP molecules. So that's yeah. Or no, per molecule of glucose.
Okay.
So that
So how come you're saying you have to use ATP to break apart the glucose with the enzymes, and then you get 2 ATP back or something, but then you take it to the mitochondria, and all of a sudden the mitochondria is able to squeeze way more ATP out of it.
well, that's because in just glycolysis, you haven't run through Krebs, so you haven't loaded up the electron transporters. But this is why, like, if you've heard of lactic acid buildup, and this would be like, that you go to the gym and the next day, like you do, 100 squats, and the next day you're like, I can't move my legs. And this is partially why, because aerobic respiration, which is what I just explained, requires oxygen, right? You need oxygen as that final acceptor. But anaerobic respiration is basically glycolysis. So it's a way to produce energy, but it's a much less efficient way to produce energy.
Hmm.
And you kind of the cell is willing to invest that upfront cost of the 2 molecules of ATP because it knows it's going to get back, let's just say 40. So like it's a worthwhile investment because you know what you're getting into. And that's what's so amazing about it is that it's this process that's occurring in like every cell of your body right now and in every cell of my body right now, except for some of the cells which don't have mitochondria, but that's a different issue.
What about, oh, sorry, go ahead.
No, that's
what about fructosis?
Fructosis?
What about the other sugars?
Oh,
Lactosis?
sucrose.
Also, what about fats and proteins?
Yeah, so that's what I was saying is that they can enter at different parts of that cycle. They can enter at different points along the chain and still come in and get used. It's just different entry points for them. Yeah. But I feel like this is kind of an information dump.
No, I think it's been good. I like this one's been I've been able to follow this one a little bit better than the last one because I can kind of like at least imagine a little bit step by step, and yeah, it's been good.
Like and this is kind of like just at the surface, but we could totally like follow it up next time with a deeper, I could really go through if you're interested.
Yeah, I mean, I'm really interested because I like it's just cool. It's really nice coming from a background, so I understand about a lot of the fundamentals of conservation of energy, a lot of entropy. How I mean, energy is similar on a macro scale. For the stuff that I've been trained in, and
totally.
And so it's really interesting to see how the energy is conserved and how but yours is just all molecular, like chemical energy.
Right.
When mine is like, what I'm used to is just, like potential energy, kinetic energy.
Right. Potential kinetic.
Gravity is a huge factor of like stored energy from here to here. And then, yeah, so that's, and then energy from the sun in form of radiation and like convection and conduction rather than energy stored from the sun in cells. So it's just kind of cool. It's cool how it does connect. And
it's just on what scale. Wait, is potential energy is PE equals MGH, right?
Yeah, because yeah, potential energy, which is just, you're basically saying how much does it weigh, and then multiply that by gravity, which is a force, and then
right.
Well, how high is it?
Yeah.
And so then you're basically, and there's other forms of potential energy. You have like if you pull a spring back.
Yeah.
Yeah.
K, right? Isn't that the spring constant?
KX is Hooke's law.
Yeah.
So that's another form of potential energy. And yeah, it's just like, and then how do you conserve energy to get what you need, and it's really fun coming listening to a different discipline and some of the fundamentals of like, well, how does this get the energy it needs to do what it needs to do? And it's like, well, we have to eat food, and then that food has energy inside of it, and then that food comes in, and it's through so many different processes it's just, yeah, it's been very entertaining. So,
It's it's truly unbelievable, and I could totally dedicate the rest of this podcast to biology, which I know we won't because it's technically the rabbit hole, but biology is a rabbit hole.
yeah, this is we can do whatever we want. You know what I'm saying?
I love this, and I would love to even. I think like something else you would find, or just people think is really interesting, is the nervous system and like how we
Mm.
communicate. And I could totally walk through like neurons and how they work and how we integrate, and then how some of this, like specifically what I'm talking about with the sodium, with the potassium, like why that actually matters and how that actually comes into play, which would be amazing to share that. And even like we just talked about the digestive system or the immune system and going through the different types of white blood cells and what they do and their jobs, and
Yeah, that all sounds so interesting, and
it's
it's also
fascinating.
fresh to me, because I really haven't done much study into this, and I've always been interested in it. So it's pretty
Yeah.
nice, and also it's good because I get to walk through it through the eyes of a beginner.
I
So
love it. I know.
like, some of the stuff that you're like, it's just, you've done it so much, you don't know where the beginning is, and so it's I think it's good.
That's why I was kind of like, we'll do, well, not that this is bite size, but like we'll do a like overall introduction and then fill in the layers, because otherwise it's too overwhelming. And if you don't have it kind of conceptually in your mind, like you can't dive deeper
Mm.
to it. But yeah, I mean, I've done this for several, like not several years compared to some people, but done it for a few years and it never gets old. Every time, like even just now talking about it, I was like,
Yeah.
yeah, this is so cool.
And it's also, this is kind of a side tangent, but just in general, it's always great to surround yourself around people that are excited and passionate about things, because it's contagious. And so whenever you're excited and passionate about this biology, it's good energy. It's like talking about energy all the time, right? And I also, that's why I think kids, because I've been around Rosemary and Judah, which is my brother's kids, my niece and nephew. It's so fun to be around kids because, well, it depends on the training, of the kid, but um, their energy is so fun. So you're surrounding yourself around people that are passionate about life, even though they're toddlers. They're still seeing everything with like lots of energy. It's really fun, and it's just contagious.
It is. I even picked that up from my sixth graders. Like the more that they've gotten kind of like they've bought into, oh, math is exciting and interesting. And then, just well, why? 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 Why does it go after zero? Why does this happen? Why is this? Why can't you do 3a minus 4b? Right. And then it's like getting into like, well, because a and b represent different things and they don't have equal numbers. And so you can't plug in
But
opposite things.
you start breaking it down, and you get into like the philosophy of math and like non-Euclidean. I don't know if that just pertains to geometry, but like, what is a point?
Descartes, what were you thinking?
I hope these sixth graders haven't gotten that far yet.
Well, they did ask me, they were like, what's the biggest number? And I was like, well, infinity, which isn't really a number, it's a concept. And they were like, what's that? And I was like, I'm so used to working with infinity, like,
Buzz Lightyear?
but yeah, that's kind of what I said. I was like, so we know Toy Story, right? To infinity. And they were like, but what is infinity? And I was like, okay, look, I love how like fixated you are on this, but let's wrap this up after class because I actually need to get through this material today.
Yeah. That's tough to know when to stop the inquisitiveness and just say, all right, we'll save that for a rainy day. That's
Even
awesome.
I though, like, you know, now you just right, like I'm sure with your math, and I didn't do all the math that you did, but like you get so familiar working with infinity. You're like, all right, some kind of concept. I don't, whatever. It's something like that. But then I was like, yeah, well, we don't really know what infinity is. It's like a concept so that we can talk about really large numbers and things that don't have a definite answer, like the limit does not exist.
Mhm. Yeah, it's a useful tool. It's a useful tool that people use to model the world. How about that?
I love that. Maybe I'll hit him with that tomorrow.
Yeah. At least in engineering, because everything's engineering mindset is, well, is it good enough to where if we do something, it's going to work? All right, well, we'll do it. That's like it's not pure math, but that is the engineering style. So,
Yeah. Well, this was super fun. I love everything biology, so I'm definitely down to spend the next several weeks of the podcast like really deep diving and exploring and
well, I'm here for it. Sounds like a good time. Uh,
learning.
and on that note, this is the end of the podcast. Wrapping it up. I'll take
We're
it easy.
going down the rabbit hole of a cell.
Yes, yes, indeed. All right. See ya.